In this video, we are going to look at viral replication. So, some of the following are keywords that you will see in the video. First of all is pathogens, host cell, capsid, RNA, genome, lactic cycle, maturation. And I want you to please take a note of the meaning of each keyword throughout the video. So, first of all, what are pathogens? Pathogens are microorganisms which cause diseases. An example of a microorganism are bacteria and virus. Bacteria produce toxins and viruses damage cells. This is how they cause their disease. Bacteria examples are cholera, typhoid, and whooping cough. When we're talking about viruses, it's usually influenza, measles, mumps, rubella, and chickenpox, just to name a few. So, how does viral replication occur? First of all, you have a host cell, and then with that cell, the thing in purple is a virus. Okay, and the virus attaches to the host cell. So, inside the virus, there is genetic information which is in the form of DNA or RNA. So this is the viral DNA or RNA. And the virus attaches itself to the cell. So first step is attachment. The next step is called penetration. And it's called penetration because the RNA or the DNA of the virus is inserted into the host cell. Okay? Whereas the protein coat, or another name for it, is the capsid of the virus, is left behind outside the cell. The next stage is replication. So what the viral DNA does is it uses the cell's organelles to replicate its own DNA. So the viral genome, or another word for genome is genetic material, is copied using the host cell, or is replicated. After this, you have synthesis. Synthesis, from the word, you should understand, is to make something. So what it does is the host cell's organelles start to make the protein coat and all the other parts of the virus. So it uses the host cell's organelles to make virus proteins. And as you can see, the next one is assimilation. So think of assemble. So the host cell physically makes the viruses, and they wait in the cell for a couple of stages where they mature, and that's called maturation. So when they mature. So at this stage you can see that you have now mature viruses inside the cell, and they have multiplied compared to the one virus that you started off with. From here, it then is released from the cell, so the viruses burst out of the cell and are released and can travel around the body, and therefore attaching to new host cells, and the whole thing repeats. How the viruses are released, there is many forms. It's not just by bursting. There is forms called exocytosis, apoptosis, and cell bursting. So for some extension activities, you can look into what exactly they mean.